to be here after such a long time with this COVID break that we had. I'm actually spending most of my time these days in Optum Labs, which is part of United Health Group. And I'm going to talk today about our work from UCLA and just kind of briefly mention what we're doing also in Optum in the beginning. The main relation between the two things is actually, and actually before I go there, the title is Whole Genome Methylation Patterns as Biomarkers for EHR Imputation. And I'll try to show you at the end of the talk that there's a kind of, is this working? Yes. That the whole genome here is what we're using, but it's not really clear that that's what we need, that's basically necessary, at least for some preliminary results that we have right now. So this is actually one of the, so the main thing that kind of we're interested in both side of with my two hats essentially, which is basically trying to find, to do disease prediction using any type of data that we can get our hands on. And it's not necessarily just genomics data. In the UCLA lab, we're focusing mostly on the genomics, but not only. But in principle, that's kind of the holy grail, right? If we basically would be able to look at the diagnosis, to take the data, get to prognosis and diagnosis of a patient at the personal level, and then understand what treatment is. And in a perfect world, we'll have all this information and actually have, in a really perfect world, we'll have all the molecules of the patient basically screened somehow and get kind of a multi-dimensional information about this. In reality, what we have is this kind of things, which is, starting from the right, because I'm from Israel. So genomics, imaging for pathology or for other types of radiology, ophthalmology, and so on, and physiological waveforms that we get in the ICU and so on, and of course the medical records, which is kind of an incomplete picture of what of the patient. So just a couple of examples for some projects that we've done in the lab in the last few years that are not related to genomics, and then I'm going to go into the genomics. So this is an example for just of what we did here, which is essentially imputing continuous blood pressure using PPG and ECG waveforms. There's actually, for these patients, we also have the genetic data, but we didn't really find a way to actually make it better. But this is something that happens when people go to the ICU or to the OR and they're high-risk patients, then we want to know what their what's the blood pressure is continuously and not just measure it every 5 minutes. And so for that, typically what you do is you do an interventional procedure, which is basically put a catheter, then you measure them continuously. And the idea here is, can we do it just with ECG and PPG? Which is essentially non-invasive. So this is one example. Another example is with medical imaging, in this case ophthalmology, which is, again, trying to basically use the data that we have. And for these ones also, we have some genetic data, but we couldn't find how to use the genetic data to improve the results so far. It doesn't mean that it's not there. We just don't know how to do it, which is essentially going into in this case, the three-dimensional imaging of the eye, of the retina, and trying to understand in what stage is the patient in terms of their age-related macular degeneration. So these are two examples, and we are thinking about basically how to include the genetics. What I'll talk about today is more on the EHR side, and basically thinking about how to use the EHR and machine learning in order to predict disease. And so we've done a bunch of works around that. And one of the steps, the main steps in all these works is trying to figure out how to deal with the fact that the data is sparse. So EHR data is sparse, and that's true for any EHR system that I've seen so far, just because patients are basically not showing up every day to the clinic, right? And so one of the things that we can do is try to do imputation, and I'm going to try to talk about this today. The tricky thing, unlike genomics, is that when you look at EHR data in the sparsity in EHR data, the missingness is not random. So in many cases in genomic data, it's actually maybe not always true in genomics, but in many cases in genomic data, we can assume that when we don't see a specific variant or we don't see, there's a no call, that's kind of a random thing. It doesn't happen because of something else that happens to the patient. But in EHR, we're not going to test, we're not going to send someone to an MRI if they don't have a problem, right? We don't, for the most part, we don't do screening. So it's not always true. There's some screening, but in 99.9% of the cases, there's no screenings. And so the fact that you don't see something, you don't have a test, that actually tells you something. That said, all the methods that are used right now, or at least most of them, there's been some works trying to kind of alleviate this, but basically most of them are essentially kind of ignoring this fact. And so, for example, basically one of the most typical ways is to do matrix completion, which essentially assumes, doesn't take into account this causality issue or this fact that you have none, that the missingness is not in random. And so the question that we can ask is basically, can we use genomics in this case to do imputation? So what is imputation in this case? It's really just prediction of what the patient has at this point in time. So that's been done many times, actually, and many people have looked at this from the genetic perspective. And the kind of most widely used or widely studied approach is called polygenic CRISPR, which is basically taking all the SNPs or the variants in the genome and summing them up linearly, so that if you see, just in this example here, you have 3 variants, the first, the third, and the 7th. So you add up 3 numbers, A1 plus A3 plus A7, you get a score. You do this for the second one, the third one, and so on. And you get every patient has a score now, which is just a linear model across the, in this case, that would be SNPs, but any set of variants, basically. Now, of course, when you do this, then it's very easy to overfit. So you basically want to either think about regularization or think about other approaches that basically take this. And there's a lot of studies on how to take into account linkage disequilibrium, so basically correlation and things. So it's not as easy as I show it here, but that's the basic idea. The basic idea here is you have this PRS that then you have a distribution in the population of these scores, and you hope that high-risk patients are going to be basically here. Okay, so one of the ways that we could basically get better and better at doing these PRS, polygenic CRISPR scores, is getting these biobanks. So this in this slide here, it's basically I'm describing the UCLA Health biobank. There's many others, obviously. Well, I'm sure people know about the UK biobank, and there's a few other institutions that basically institutions that built biobanks of this order of magnitude, or even more. In UCLA, basically, we have 150,000 people in the, it's called Atlas, the biobank. What we have over there is we keep the samples, the DNA samples for each of the patients. So we don't keep the entire, the blood sample, but we basically extract the DNA. And that's important because if you wanted to do now an RNA study on this population, that would require another collection. While if you want to do anything related to the DNA, for example, methylation, you basically can use just these samples. And that's essentially what we did. So some limitations of the PRS. So the first one is pretty obvious, which is basically the genetics doesn't capture environment. So that essentially means that whatever I can predict about a person now, I could have predicted it also when the person was born. So if I want to ask what the patient has right now, it's kind of, it's maybe important to understand what is the kind of total risk of the patient to have a heart attack sometime in their life. But I don't know necessarily, unless I have unless it's the age of onset is very heritable, I don't really know exactly what's going on right now with this patient. Okay, so that's one limitation. I guess the second, I guess I mentioned the second one here. The first one is, okay, so the first one is basically mostly an environment, which is essentially if someone smokes, I wouldn't see it. The second one is really the fact that it's static. And then the third one is basically that if you just look at the kind of the proof is in the pudding, PRS is not really used by clinicians today. And it's not that we tried it only for a year or two. We basically, I mean the community, basically, we've been looking at it for many years now. And still, if you look at the clinic, it's not really used. So there could be other reasons. It's not necessarily that the prediction is not good, but if the prediction was excellent and it was very useful, then probably we would be using this. So then what we ask is, can methylation be another approach to do kind of a PRS or MRS, methylation risk score, where we basically are trying to do essentially the same thing. So methylation, for those of you who are less familiar, basically is a biomarker. These are modifications in the DNA across the entire genome, and every cell. So unlike genetics, basically genetics, so DNA. So when you look at the genetic data, every cell has the same genetics, roughly, almost. In methylation, essentially every cell has a different one. There are some correlations if they're the same, if they're similar cell types and things like this, but basically every cell is going to have a different methylation pattern. And there's been a lot of work over the last few decades, I guess, showing correlation to diseases and the fact that it captures demographics. So for example, you can basically, there's differences, major differences between women and men. Even when you look at the autosomes, it can capture ancestry. So there's and it captures really well age. So basically Steve Horvath from UCLA basically did work, which is I think maybe 10, 15 years now, where you could show that you have a very high correlation between age and methylation patterns. The other thing is that it's inexpensive, although today everything is inexpensive because you can do the genome for $100, but basically it's been inexpensive for a long time now, for the last 10 years, at least. So the data for methylation basically looks kind of like this. In every, if you think about a matrix of individuals, you'll have individuals and methylation sites, and you'll have a number between 0 and 1 that will tell you what is the frequency of the methylation in the samples that you're looking at for that individual at that position. So these are positions in the genome. Typically, people use these methylation arrays that so one we use basically has 800,000, a bit more than 800,000 positions that we're looking at. Okay, so then the question is, can we use, instead of genetics, can we use methylation for imputation? And so that's what we're trying to answer here. And so why is this promising? So methylation actually does capture some of the environment, maybe a lot of it actually. So for example, smoking would be something that you'll be able to find using methylation. So to at least some correlation to how much people smoke. And there's essentially basically everything that happened, not everything, but a lot of things that happen to the patient, which are not necessarily something that they were born with, are going to either change the methylation or the methylation is going to change that. Either way, it doesn't really matter the causality, there's going to be some correlation. The other thing is that methylation is dynamic. As I said, basically you can capture age, for example, so it changes over time. It's actually nice. Methylation is a nice biomarker in the sense that it changes typically over months and not within hours, not within days. And so that basically means that you can capture kind of the information about the patient over the last few months, if you think about it intuitively. And then, unlike PRS for genetics, methylation, if you look at all these biobanks, they essentially don't have any methylation data. And so there's no, it hasn't been really studied. And personally, I don't really understand why, but hopefully I can convince you now that maybe now we should start doing this. So the basic, what we tried is very simple. We tried to look at, instead of a PRS, which is just a linear sum of genetics data, now we're looking at MRS, the sum of beta I MI, which is MI is the methylation at position I. And then we measure this over 800. So that 800 is after some cleaning and some pruning, but basically 800 patients from the Atlas population. So we had also the genetic data, and we had all the EHR data for this population. This population actually that we took these are people that went to surgeries, and we looked at the risk for AKI, acute kidney injury after surgery. So they all had some kind of, and that's how we matched them basically. And they all had some issues that are related to the kidney. And actually some of our best results are basically in predictions of kidney problems. Okay, so, and then the method is very standard. We basically just used regular regression. We tried multiple versions. So ridge, lasso, elastic, and then we also try to combine the genetics and the methylation. So everything that you could think of as kind of reasonable to try. And we compared it to a baseline, which is basically regression over these features here, which is the kind of demographics, some information about, so BMI, and then cell type proportions, which is an important fit that actually changes the methylation patterns globally. The ancestry, smoking status, and 10 top PCs of the genetics. Okay, so that's our population. So again, 800 people. We had some diversity, but still 70–80% of the people were Caucasians. Our goal originally was to basically just look at Latinos, but we just didn't have enough people that were kind of matched everything that we needed. So we ended up with this kind of mix. And the populations are a little bit on the older side. So basically they're skewed to the right a bit. And the reason is because these are people that basically going to surgery. So they actually visited the hospital for a specific reason. Okay, so these are the results that we got, just a summary of those. And what you can see is, so I'll give you just an example. So we looked at about 600 different parameters in the EHR, including medications, Phi codes, which is essentially combinations of or groupings of ICD codes, so diagnosis codes. So these are, you can think about it as disease, and labs. And every point here is one of these labs or one of these diseases. And what you see on the x-axis is what the baseline gave, just a simple regression on just these 10 features. And the y-axis is what we got with the PRS or with the MRS. So a point that is on the diagonal is basically not doing better than just the baseline. And what you can see is that the MRS gives you, in many cases, not all the cases, but in many cases, you get things that are above the diagonal. So it's pretty significant. And it's only with 800 samples. So we didn't do 150,000 or 500,000 like the UK Biobank. It's a very small number compared to what we're used to with genetics. These are specific examples where you can basically see for these specific phenotypes, what happens when you look at the R square of the genotypes compared to the PRS compared to the MRS. And these are basically just combining those into the, predicted deciles compared to actually what's the fraction of the cases in each of these deciles. So ideally, in an ideal situation, you'll have everyone on the right side, right? So, and you can see that there's more of a push of this in the methylation case. So if I just take the top 10% in the methylation risk scores, I'll get that 70% of the cases are actually here. While in the PRS, it's only going to be 40%. Okay, so I have a few of these slides. I'm going to go through quickly through a couple of those. So these are just some examples for some of the kind of highlights, some places that just kind of to show that in some cases it was very substantial, the differences. And you can see that some of these things are related to kidney by definition, or kind of by the bias of what we, a centennial bias, I guess. But these are true results. It's just basically we had more power to do it when we look at kidney related issues. Okay, so one of the things that basically we asked ourselves is that people are actually using, when they look at imputation, if we that was kind of our original goal, they look at matrix completion, and we're now coming up with, we're comparing to PRS, but nobody's using PRS, even though we would like to say that people would. People don't use it because it's not there, the data is not there. So people use these matrix completion methods, and the question is, can we add to the matrix completion the methylation, and how much do we gain by that? So what we did is a very simple thing. We took basically the matrix, which is the kind of with the missing data, and we added the methylation risk scores, kind of a matrix on the side. And so now we have a much bigger matrix that basically half of it essentially has some data and half of it has missing data. And then we just basically put it into the matrix completion. Obviously, this is not the optimal way to do this. It's actually a pretty lousy way to do this, but we just wanted to see how that works. And it seems like basically just even that actually gives you a pretty nice improvement in performance in many cases. So again, not always, but I'm pretty optimistic that if we try to optimize this, we would actually be improving some of these results as well. Okay, I think I have, what is it, 8 more minutes. So yeah, so this figure here basically shows that, just kind of, I'll give you just the gist of it. It basically shows that if you try some of the things that we try to predict, let's say an ICD code, so a diagnosis code, maybe happened 6 months before we actually took the methylation data. So you can ask yourself, how well do we predict something that basically a lab or a medication or anything that basically we try to predict, that the event happened 6 months ago, it didn't happen right now. So obviously, if something happened 5 years ago, then your chances are typically much lower. It depends, actually, if it's a disease, maybe it continues. But if it's kind of a lab that maybe you had a spike or something like this, you shouldn't be able to see this. But we actually see that for the most part, what we get in terms of the prediction when we go back a year, it basically gives us very similar results. So the results are maintained even if you go back a few months. Okay, and the other thing, we had, again, some diversity. So we compared basically the results across the different relations. As you can see, the confidence intervals are huge. And the reason is because our samples are small. It's only 800 people. And so I don't know if you can basically conclude that there's no differences in methylation risk, because in PRS, there are actually differences between when you look at different populations, but there the numbers are much larger. So maybe if we go to 100,000 and then we do the methylation risk score based on 100,000, we'll see differences. But at least with these sample sizes, we don't really see differences. So just remember that we had like maybe 100 or 50, between 50 to 100 African Americans. Okay, 5 minutes. Okay, so and again, for those of you who are interested in building biobanks, I know some of you are. If you look at these figures here, and you basically try to kind of sample the data and say, instead of 800 people, if you only had 400 people, what would be the result then? And instead of 800, if we had 200, you can basically try to see how how things are basically going to change over time. So in this case, if you look at the baseline, for instance, it doesn't really matter. So the baseline is just taking the demographics and the BMI and so on. So it doesn't really matter. So if you took 800 or 400, it's essentially the same results. But if you look at the methylation risk scores, in some cases, it seems like we're just in the beginning. So if we went from 800 to 8,000, probably the results would be way better. Okay, so I'll just finish with a couple of things, and I put this picture for Roderick here. So this is a work in progress. That really kind of fresh out of the oven, so still maybe buggy, although I think it's pretty safe. What we try to ask ourselves is basically, so what we did so far is just a simple methylation risk score using linear models. So we tried all kinds of linear models, but the question is, can you actually use nonlinear models and get better results than just looking at the linear models. And so we have this this nonlinear model, which is essentially a neural network, which is built this way. We have a set of genes. Actually, in this example here that I'm going to show, we took 30 genes, and these genes that we chose, so you could do it across the entire genome, but so far what we did is we took genes that are associated with the condition. And so for each of these genes, you build a neural network, which is in this case a fully connected neural network, relatively shallow. You don't want to add too many parameters because then you're going to overfit very easy. And then you get essentially a bit or a score from each of those. So a number from each of these genes. So now if I have 30 genes, or if I have 20,000 genes, I'm going to have 30 numbers or 20,000 numbers, depending on the number of genes you're going to look at. And then you basically put all of them into another fully connected neural network that includes these numbers plus information about demographics, the genotype disease, and the cell type composition, kind of like what we had in the baseline. And then we tried either adding the MRS or not adding the MRS. So you may be saying, okay, let's try to add MRS and see if it actually helps, or just basically let's just ignore it and see what these 30 genes give us. And so that was what I was hinting in the beginning. So this is these are the results that we have so far. So random forest actually gives you nothing. If you do just ridge across this population, so these genes, that's what you get, about 0.6. And then if you look at the neural network without the MRS and the neural network with the MRS, they're actually doing about the same. So in fact, in this case, 30 genes gave you the information from a predictive perspective, which is essentially almost, or I mean, it's hard to say because again, the numbers are maybe too small if there's a difference, but definitely comparable compared to the other approaches. Okay, so these are again work in progress, and we've only done it on a couple of examples because for each of those we had to go and look for the genes. It's like it requires some more work, but I think it looks promising in the sense that kind of the non-linear models. If you just looked at the linear model, so there's two kind of messages here. One of them is if you just look at the linear model, that's not going to get you to where you want to be. So the non-linearity actually helps. And the other thing is that between these two, maybe we don't actually need the entire genome. We need to basically choose the parts of the genome more kind of wisely, and in this case, using the literature. Maybe there's another way to use the entire genome, but at least right now, we don't really see an advantage for this from these examples. So, okay, so conclusions. I have like 2 more minutes, or I'm done. 1 more minute. Okay, so just basically conclusion. So I'm thinking methylation. That at least hopefully I was able to convince that methylation can basically give a good signal. I'm going to risk it and say that hopefully we could basically find a universal blood test, and I'm kind of in the bad blood just recently, which is it's kind of scary to say that, but basically, so not Theranos, but really universal blood test in the sense that it will give us some idea of basically what's going on with the patient without actually doing 1,000 things to the patient. So there's many different things that you could do to try to improve. I mentioned a couple of them. So increasing the population size, that's an obvious thing, right? So basically, and I think that's what I would urge everybody that is working on these biobanks to think about doing. Even if you go to 10,000, it's 10 times more than what we did, and it's not so much money today. Non-linear models, as I said, and improving the essentially the matrix completion. We use a software called SoftImpute, but basically you could use others for matrix completion. And then from a biological perspective, what we showed are just correlations, and it would be interesting to understand what is the kind of causality, what causes these predictions. And so basically some type of mediation as a causality. And then another idea would be to basically look at cell type specific, because again, we know that methylation data has this kind of composition of all of these things together, of all of these cell types together. All right, so thank you for your attention. And this is basically based on, most of it is based on the paper that Mike Thompson from my lab, who's actually going to Barcelona next year, and Brian Hill from my lab basically led. And then Eran did the nonlinear models that I mentioned just now. And then the others basically helped as well. Thank you.
Thank you, Eran. I think we can do one quick question before we all go for coffee break. Yeah.
Thanks, Eran. It was really nice. I was wondering, did you already, before you do mediation and causality, just break down the features that were picked up in the methylation array into tagging, cell type composition, the methylation clock? There's lots of knowledge about which features are tagging specific properties of a sample. And did you already do that to get an insight of what you actually learning?
No, we haven't, because what we did is kind of a screening across everything. And to do this, you would basically have to go to each one of them specifically, because unless we think that there's going to be something universal in that sense. My intuition would be that for age, there's going to be specific mechanism, and for type 2 diabetes, there would be.
You could just break down the feature relevance, in these broad categories for different traits.
I think that would be super important, and no, we haven't done it yet.
Let's thank our speakers again, and if you have questions, let's move the discussion to the coffee break.
All right.
Thank you.
Thank you.